Thanks to Nanopore for the invitation to speak today. I'm going to talk about how we are using Nanopore sequencing in my lab to answer some questions in the research environment, some clinically relevant questions that we come across at Moorfields Eye Hospital. So a little bit of background. So we are, as was mentioned this morning, we're in the midst of a genomics revolution now, and this kind of came about in 2009 with the advent of the NGS era, and so this culminated in the UK with the 100,000 Genomes project, which again you've heard about earlier today. But the diagnostic yield of whole genome sequencing using Illumina technology in the UK is somewhere in the region of 25% for rare disease. We do a little bit better than that in ophthalmology. We got up to 55% in the pilot study for Genomics England, and this is because we understand the genetics of rare eye diseases very well. We're very good at deep phenotyping these cases, so we often have a really good clue as to which gene that we are looking at. So now, genome sequencing is available as an NHS test for many rare diseases, including retinal dystrophies. And so what is a retinal dystrophy? It's a leading cause of blindness across the globe. It affects somewhere between 1 in 2 to 4,000 individuals, and it's caused by dysfunction or degeneration of the neural retina or the RPE cells that support the neural retina at the back of the eye. Molecular diagnosis is challenging. There's a very, very broad spectrum of phenotypic, genetic, and allelic heterogeneity, many genes described to date, spanning all inheritance patterns. And so, why does whole genome sequencing not work all that well? So the short reads lose genomic context. So we don't understand how to map them very well. They're too short. So we can't map them to repetitive or low complexity regions. Pseudogenes, as we've seen today, are not well covered by short read sequencing. And there are regions of the genome that aren't well characterized by short read data as well. Another big problem for clinical labs is that of variants of uncertain significance. So VUSs are a huge problem. They represent a vast number of variants that are identified in clinical samples, and we don't know the implications of those. Are they pathogenic? Are they just benign variants that are rare? How do we better understand what these variants are doing? And one group of variants that we think is going to be important are those intronic, missense, and synonymous variants that may actually be affecting splicing. But we're interested in the retina, and transcript analysis is very difficult because these genes are only expressed in the retina, and no one wants to give a retinal sample in clinic. So that's one big problem for us. And then again, uncertain genotypes, being able to phase distant variants in genes in recessive disease specifically. And this is the biggest problem for a gene called ABCA4 in retinopathy. This represents about 30% of all inherited retinal dystrophy cases are accounted for by this one gene. And there are many different variants. So I think over 2,000 variants have been reported in this gene now. Okay, and here's another problem gene that we have. So this is the opsin array on the X chromosome. And this is what governs color vision. And rearrangements and mutations in this gene, they cause red-green color blindness, but they also cause severe early onset congenital cone dysfunction, cone dystrophies in affected males. And there can be up to up to 10 copies of the opsin genes in an array on the X chromosome. And what this means essentially is that when we try and sequence this region with short read sequencing, we can't map any of the reads. It's a complete black hole for Illumina sequencing. So to overcome this problem, so I'm going to talk through a few examples of how we are overcoming these issues in our lab now. So to overcome the problem of sequencing through the opsin array, or large regions and low complexity regions, we turn to this method called catch. And what essentially this does is it's a method to to maintain the integrity of high molecular weight DNA. That's all it does, and these figures illustrate how that works. And so what we're able to do is we're able to cut out our region of interest, so the opsin array, for example, which can be up to 250 kb on the X chromosome, hold the integrity of that, and then sequence through that region with ultra-long read sequencing. And when we do that for individuals who we're interested in the X chromosome for, this is what we get. So the Illumina data at the top, and then the Nanopore catch long read sequencing at the bottom. And we get full-length reads across the entire array. We're able to count the number of genes, the opsin genes that they have. We're able to haplotype in females who carry 2 different haplotypes across this region. We can detect mutations in the opsin genes, and we can characterize those mutations, find which gene is affected, which is also key for this disease. And that's what it looks like when we zoom in a bit closer. We've got really nice, even coverage across these 2 exons in the medium wave gene, and we can detect single nucleotide variants. I think our record read depth for this is about 120x so far. And so we use this similar method for ABCA4 to try and phase distant variants then. So we've done this now with ABCA4. We can generate good read depth across this gene, up to about 60x across this gene. We get full-length reads across the gene. We can phase any variants. We can characterize all of the haplotypes, and we're able to feed back to individuals the phase of distant variants when family members are not available for segregation studies in clinic. And this is an example of that. So this was an individual who carried 4 rare variants in their ABCA4 gene, and we're able to phase and demonstrate which of those variants are in trans without access to family members. And so the last thing I'll talk about then is these variants of uncertain significance. So these are some examples of intronic and upstream variants in retinal dystrophy genes that we suspect are affecting splicing. And we're able to characterize these variants in our lab now with long read sequencing, with nanopore sequencing, not necessarily long reads for this application. And we think variants like this are responsible for somewhere in the region of about 10% of the disease alleles that we're seeing. So how do we do this? So we're able to predict the effect of splice variants using Splice AI, and it's very good, but it's still just a prediction. It's not proof of effect. We need an assay to be able to prove the effect of these. But like I said, in the retina, these genes are only expressed in photoreceptor cells, so we can't traditionally detect transcripts of these genes in blood. So research labs will turn to things like mini-gene assays or iPSCs to characterize splicing effects. But this is expensive for clinical labs to take on. So nanopore sequencing is a bit different. So we aren't relying on a signal strength high enough to be able to detect an electropherogram or an agarose gel with nanopore sequencing. If you've got one molecule of your transcript in your tube, you'll get one read from that transcript. And so we're able then to capitalize on that and use nanopore sequencing with RT-PCR for our specific non-expressed genes and characterize the effect of those variants. So this is an example from a paper that came out last year where we had this minus 25 variant upstream from an acceptor site. We're able to detect a shift in isoform usage to this non-coding isoform, and we're also able to phase that shift in isoform usage onto the allele with the mutation versus the wild type allele which carries a coding SNP. And this is something that we actually see in controls. We see very low level of this isoform usage, but it's very much increased in the mutant allele specifically to about 90% of the transcript that arises from that allele. And so just to take that a little bit further, we can use this same method to detect splice variants, splice alteration in genes that aren't expressed outside the retina at all. This is a gene called TOLP1 The discovery of this goes back to 1998, and this mutation was actually in that discovery paper. And we're able to finally characterize the effect of that mutation using nanopore sequencing, and what it leads to is a frame shift, an alteration of splice site usage in exon 15. And so just to summarize, there is still significant missing heritability. Short read sequencing is limited in how it can characterize variants and detect variants, and we think that our methods that we've shown here represent the potential use of nanopore sequencing for clinical labs to be able to detect and characterize this sort of variant. And I'll thank my collaborators and colleagues and my funders, and thank you very much for your attention.